Hello, everyone. My name is Yuzhen. I'm me plus tons of bacteria. So many of them are good, and many of them are bad. I don't like the bacteria on my teeth because they create a lot of problems to my teeth. But anyway, I also have a lot of good ones. So my lab, we have been trying to develop new algorithms, new tools, so that we can study those bacterial communities a lot better, faster, or efficiently. So that's reason I and I'm pretty sure many other scientists were very encouraged by this announcement from White House of a national microbiome initiative. So it's really about how we can study those microbial communities from different ecosystems, not only those ones associated with human beings, but also whatever bacteria you can find anywhere, floor, on a keyboard, anywhere. So bacteria, they live everywhere, and they are very important. They play very important roles on human health, and sometimes they cause disease, but a lot of times they're very important for our health and also for the health of the environment. So we really need to take a very careful look at those bacteria, those micro tiny organisms that you don't even see them. So yeah, that's as I said. So they're very important for the health of human beings. And sometimes, if you're happy, your bacteria are happy. And if your bacteria are happy, you will be happy. So we know a lot of things are happening, although we know some mechanisms, but we don't know everything. So that's really scientists are trying to figure out what those bacteria do and why, how they respond to host changes, the environmental changes, and so on. So we have been developing new algorithms and the software so that we can analyze the data from those microbial research. So those projects try to determine the DNA sequences and other types of information from a microbiome sample, just communities of bacteria. So we have developed quite a few methods. Some are more successful than others. So we try to, a lot of those data from microbiome research, they're very complex. In terms of volume, they're huge. So some of those huge microbiome research or projects, they produce like terabytes of data. And those data are really from a mixture of bacterial species, thousands of bacterial species. So this is a computational very challenging. So we have been trying to develop new methods and tools so that hopefully we can help others and ourselves to interpret those data a lot better and more efficiently. So those are kind of generic approaches like gene prediction from those from the genomic sequences from microbiome samples, or a lot of time we do a similarity search, like you do Google search, but we do look for similar genes, like if this microbiome contains genes that are similar to the known ones, or encode pathways that are known for some reasons. So similarity search is one of the key technique or key thing that we do. So because of the volume of the data, so this is really has become a very challenging problem. So we also try to come up with new methods to help with those similarity search. The tool we have is called Rep Search 2, so it's fast. Although there are some competitors coming out there, but that's our goal. We really try to come up with better methods, and also we try to come up with like new ways of mining those data from different angles, perspectives. So we call like target approaches. So we target those important, evolutionary, functionally important genomic elements that we can find in those microbiomes. So and also we try to integrate the heterogeneous data from those microbiome projects. So this is just one example of how we do this target analysis or develop target approaches for those target analysis. So this is really microbiome plus CRISPR-Cas systems. So it's really, those systems are becoming the most successful genome engineering tools. So you can essentially change the genome of human beings or alien genomes using that type of technique, CRISPR-Cas, but those systems are naturally found in bacteria. So just scientists learn from the bacteria. They're awesome. So what we have been trying to do is really we develop new methods to help us to study those amazing systems in microbiome data. So that's me. Take good care of your bacteria. Thank you.